Hello, everybody. Welcome to Loop and Learn's T1D speaker series. Today is interesting. We're going to be hearing from Charles C.C. King, PhD, from the Diabetes Research Connection in San Diego, California. Before we get there, let me just quickly say Loop and Learn has a website called loopandlearn.org. Bookmark it, check it out frequently. If you have questions before you ask, you might want to look it up in there. It's really an amazing, amazing website. And there is a link to under resources to sign up for the newsletter. I implore you to sign up for the newsletter because if you are on Loop, you will get notifications if there's something you need to know about. And that's the easiest way we can communicate with you, and it doesn't cost anything. So please sign up for the newsletter. An upcoming event in 2 weeks is Anton Beckman and Elcida Rosa. Anton founded Booplan that makes bubble technology, which pulls information from the Abbott Net Libre CGM and is a part of the Diabox system. And he's also had an islet cell transplant. He's young, he's in his 30s, and he wants to share about that as well. It should be really, really interesting event. Today I have two co-moderators. I have Rachel Gerlin, who you met last week. She's a medical writer at Insulet Corporation, recently graduated in 2019 with her PhD in biomedical engineering, research in beta cell replacement. Her sister has had T1D for 20 years and is a DIY looper. And Tina Hammer is a wife and mom and former biotechnology research scientist. I didn't know that till today, who hates that her now 11-year-old son, Reese, developed T1D 2 years ago. I totally feel that one. But she enjoys the science of the T1D physiology and research. And by the way, Reese produced an amazing introduction to looping video, which you can find on the YouTube channel. And I think it's now in chat. So it's adorable. It's wonderful. If you get a chance, just watch it. So today we get to hear from C.C. King, who is board chair, president of the Diabetes Research Connection, and is a research scientist and a pediatric diabetes research center, Department of Pediatrics, UCSD researcher. The Diabetes Research Connection is interesting. It facilitates rapid funding of peer-reviewed discovery stage investigations, giving promising early career scientists the opportunity to challenge the frontiers of type 1 diabetes research. The donors are empowered to support research projects of their choice and interact with young researchers whose curiosity and vision will help define the future of diabetes research. It's a really early funding source for real promising and interesting science. Dr. King was one of the 8 UC San Diego scientists to be awarded significant funding from the California Institute of Regenerative Medicine, receiving over $1.3 million to study the role of microRNAs in human embryonic stem cell differentiation, hoping that a better understanding of stem cell differentiation will lead researchers to promising type 1 diabetes therapies. You do not need to understand what that is. He will explain it to you. He's involved with some very, very interesting research. And we're just lucky that we have his time today. And I'm going to unshare and turn this over to CC. If I can do that, I will do that. And I need to make him go. Hang on. Let me unshare, stop share. There we go. And CC, let me make you co-host. And okay, you are good to go.
Okay, hold on. Let me share my screen here.
Do you have a big dog?
I have a number of big dogs who I'm trying to keep out of my out of this room. So you will see them probably come in periodically throughout this. This is, how the home works. Are you able to see my slides?
Not yet.
Hold on. I think I've got a still spinning circle.
We know spinning circles.
Yes. All right. Can you see?
Yeah.
Okay, fantastic. So I'm going to probably ramble around a lot of different topics today. And I will talk a little bit about a question that somebody who, I'm not sure who it is who's on this call, asked about, which is why is the FDA so slow to approve new drugs? I'm going to talk about DRC, what we do. I'm going to then also talk a little bit about some of the projects, because that's really what we're interested in, is the science. And at any point during this, feel free to raise your hand, ask questions. I want this to be a back and forth. Certainly, if I was to present just all of this straight out, it would take nowhere near our time. So I encourage a discussion on this, and chances are I won't know too much about what it is that you're asking, but I'll do my best.
CC, we generally collect everything in chat, and then we volley them to you so we don't interrupt your flow.
Oh, well, I actually want the flow to be interrupted, and also I'll try to make sure to check periodically if there are questions.
Okay, that sounds great.
That works. So let me tell you a little bit about myself. Joanne did a great job introducing me, but that's a little bit in my former life. For the past 3 years, I have been at the Genomics Institute of the Novartis Research Foundation, or GNF, in San Diego, which is part of the large drug company, this large Swiss drug company, Novartis. And I work with a team in functional genomics there. And our task is to really take on a number of projects across everything from oncology to chondrogenesis to, basic biology questions. And really what we do is we do a lot of genomics work. We have libraries and screens for everything from CRISPRs to CRISPR-RNAs all the way to, cDNA collections of the human genome. So my team curates this, and we build plasmid and viral vectors. We're also very invested in identifying targets, drug targets, as well as mechanisms of action. So knowing that I've come from academics, UCSD, where I was for almost 20 years studying human ES cells, working with Alberto Hayek, who probably many on this call know. Here in San Diego, we were the first group in San Diego and at UCSD to get human embryonic stem cells and work with them in the capacity of trying to develop cell-based technologies for type 1 diabetes. So I'll kind of bounce back and forth between my science role and what my role is at Diabetes Research Connection, or DRC, in this. So feel free to stop and ask, and we'll just continue until people run out of questions. So the first question that someone asked me was, why is the FDA so slow to approve new drugs? And I thought that it was probably a pretty good set of questions to begin with, because we're all very interested in how people can get better access to more efficacious drugs, especially in the type 1 world. This is a very forward-thinking group, and a group that is very much interested in pushing medicines to people, which is really important. So I thought I would begin by going through this process and spend a couple of slides talking about what it is that goes on for the FDA. So I'm going to try here to advance my screen. There we go. Okay, so can everybody see my laser pointer now?
Yep.
Okay, fantastic. So here's the FDA kind of general pipeline. And that is, it takes 2 to 5 years of basic research to be able to get to something that's considered a phase 1 kind of idea, something that the company or any other place considers to be phase 1. And then there's rigorous preclinical testing that goes on for at least 1 to 2 years to determine whether this can go into animals. After that, then you begin really the human trials on this. And this is the clinical trials are all going on while people are still doing basic research normally to consider what the target is, what the mechanism of action is. And there's 3 phases to this. And I think many people are probably familiar with this. Phase 1 is testing in healthy subjects. And this is just to test. This phase 1 is just to make sure that whatever the drug is that you're using and are trying to develop is not something that will have immediate adverse effects. Okay. Then you start phase 2, which is testing in individuals with the disease. And that normally takes about 3 years. And that's not necessarily looking at efficacy, but looking at what happens in the patient population itself in response to this drug. And then there's phase 3, which is larger scale testing within the individuals with the disease. And I'll get into these points each a little bit more as I talk about this. And then after you complete phase 3, there's normally about a 6-month to 2-year wait before the FDA approves the drugs. And that's normally to allow for looking at data that comes out of these larger scale tests to try to find any adverse indications. Let's see. Now that I have this on, I can't do this. All right. So how do these phases work and how many people are involved in them? Which is really, I think, something that a lot of people don't fully understand. Once the drug gets past this preclinical phase that allows for the people to say, hey, let's move this into patients. This phase 1 period, which is normally about 1.5 to 2 years, is really very small. It's healthy subjects, and it's normally fewer than 100 people that have this, all right? Phase 2 is a little bit more, and this is, again, testing in individuals with the disease. And this really is not that large of a sample population, which is 100 to 300 people. And then within phase 3, when you start the phase 3, this is when you really begin to ramp up. And here you're looking for anywhere from 1,000 to 3,000 to even more patients. And this is really the data that the FDA begins to look at. It's after the completion of this phase, which is why this 6-month to 2-year period is because they want to follow a large number of subjects to get a good enough sampling of of how the drug acts. And then phase 4, which almost no one hears about, is normally lots and lots of participants after the drug has come out. They follow people who are who are taking the drug to continue to develop data to look for additional safety and potential adverse indications. Now, one of the things that I thought was pretty remarkable is this graph here, which spans from about 25 years, a little bit less. And that is, what is the success rate of any drug that's introduced here at phase 1 actually getting FDA approval and launch? And here you can see that the success rate is ridiculously low. With the exception of 96 to 99, you're looking anywhere from about 8 to 10% of any drug that goes into phase 1 actually making it to a successful launch. So, given that this is a very rough and difficult path, you can understand a lot of hesitancy that drug companies have to begin to do this. So I'm going to continue on with this thought and try to give you a little bit more information. And I found this slide particularly interesting. And this breaks down a little bit better of what's happening in each of the phases. So phase 1 and 2 are really learning phases. And remember, we're looking here anywhere from about 3 to 5 years that you're learning what's going on with the drug. you have healthy and well subjects, refractory patients in oncology. Oncology kind of gets a little bit separate of a thing because of compassion and time. They're looking at safety, tolerability, pharmacokinetics, biomarkers, proof of principle, what's the maximum tolerated dose. So this is really kind of saying how much of the drug can healthy people handle. So this is then happens in phase 2. And what you wind up with is patients that have the disease and they tend to have restrictive inclusion-exclusion criteria. They're bound by, at this point, to meet objectives. Phase 1 is basically, does this compound that we're injecting in you or biologic kill you or harm you? Phase 2 is, well, does it do what it says that it's going to do? Or does it look like it will? This is the proof of concept. This gives you dose ranging, guidance on dose regimens. And for oncology, and again, oncology is a bit of a separate beast. Phase 2 sometimes results in early approval. If it shows promise in phase 2, they will make drugs, the FDA will make drugs more readily available to patients with cancer because time tends to be of the essence. In our world, when we're thinking about type 1 diabetes, there are a number of what I like to call temporary patches and fixes that allow for people to continue to live and, in my opinion, suffer with type 1 that is precluded in this type 2. They want strong phase 3, phase 4. And so what's in phase 2? Again, patients with less restrictive inclusion-exclusion criteria, this is a longer duration. What happens instead of after the first 6 or 8 months? What happens over a couple of years? You want to get confirmation of the benefit risk profiles. You want to characterize subgroups within different benefit risk profiles. And very importantly, you want to confirm efficacious dose, okay? Now, phase 4 is looking at any new indications. Is there anything that's off target on this? Are there other new formulations, modified releases, further characterization of study, and then possibly, like with things such as the Pfizer vaccine and Moderna, development in pediatric population. So I've got one more slide on this question, and then we'll open it up because I do see in the chat that there are some questions. Okay, so let me kind of give it from a different point of view that people don't think about, which is from the drug company's point of view. So developing a new drug takes about 10 years and about $2.5 billion is best estimate. And if you want to see really something remarkable, only 1 out of every 10,000 discoveries really make it to market. So this is an incredibly low hit rate. And I want us to kind of keep this in mind as we are discussing what it is, the science that Diabetes Research Connection funds, as well as what these young investigators are trying to do. Because I definitely have some thoughts on this, okay? Now, going down to the middle here, the public doesn't understand the benefits of the clinical trials. And this is fairly remarkable, I found, is that 40% of surveyed adults didn't understand what these clinical trials were for. And furthermore, they thought, the public, indicated that patients in these trials were, quote, treated like guinea pigs. That is a very dangerous misconception and really lends itself to the problem shown in the bottom panel, which is 11% of all trials fail to enroll even a single patient. So you could have a drug that's ridiculously efficacious, but if you can't find the people to enroll in these trials, it will never get its shot. And what's even more remarkable to me is that almost 40% of all clinical trials under-enroll. This is truly remarkable for what it is that we're trying to do. So when one talks about why is the FDA slow to do this, there's numerous points along this ladder and this timeline that do it. But one of the main ones is enrolling people and better educating them. Let me just again show one more to finish off this section. Reasons for clinical trial failures by phase. And I found this to be pretty interesting. At the beginning, safety and efficacy are about 30% of the failures. One of the main ones, though, is commercial viability. Is there really a need for this? And this is the largest reason that phase 1 drugs fail, is that there's no seen commercial viability for it. Now, as you move into phase 2, efficacy takes over. Does the drug actually do what it says that it's going to do? Safety concerns are still definitely there, but they tend to drop very low. And commercial viability remains to be a large proponent or portion of what is actually canceling these drugs. And finally, in phase 3, efficacy. Efficacy turns out, again, to be the main thing. We just don't have good use of how this actually works in patient populations. And part of it is enrolling patients, but part of it is we don't understand human biology as well as we'd like to think that we do. So at this point now, I'm going to switch topics, but before I do, I see that there are a number of questions and maybe we can go through those. Joanne, is this something that you moderate? How does this work?
Sure. We can moderate. I'm going to throw it to Rachel. She wasn't prepared, but I'm going to throw it to Rachel.
Oh, no, that's fine. So thank you so much for talking to us about clinical trial research. I think it's great that we can get some more visibility on this. And as you are aware, and I think most of us are aware, we saw record-breaking FDA approval processes with COVID vaccines. Do you think, since there's a precedent that we can do things quickly, if there's enough need for it, that this will alter timelines for other drugs and devices in the future at all?
Boy, you really hit on a very important topic. So let me give you a tiny bit of the background on lipid nanoparticles and what's being used, at least in Moderna and Pfizer. And that is, the technology for those has been around for a very long time. In fact, one of the main things that I've done for the past almost 30 years in science is transfect cells using these lipid coatings. So this technology is not at all new. Now, using this technology in humans has been shown to be efficacious and really without any problems for 15 to 20 years. The FDA drug its heels on approving the technology because there really wasn't a need to do it. They, in my opinion, they spent an awful lot of time waiting until there actually was a need. And once the need came in, then they said, well, fine, we'll go ahead with it. If COVID hadn't hit, I'm fairly certain that using these delivery methods would still not be widespread use. But now that the floodgates are open, there are an awful lot of people who are trying these for everything from, Ebola all the way to cancer therapeutics. So it's a little bit of a misnomer to think that the FDA has been dragging their heels on this technology and that it's really turned around fast. What it turns out that it is more of a recognition that they have to do it. They were backed into a corner.
All right, thank you. And can you speak a little bit about how this the drug approval process might differ from the device approval process?
I am not an expert on device approval. So I know that the device approval people tend to, at least from what I've seen, be a little bit, in my opinion, more forward-looking and ready to adapt technology. However, that said, there's comfort in knowing what has worked traditionally and not a huge appetite for expanding what they define as their technologies that work. I think there's a lot of hesitancy because things have remained unproven in large-scale clinical trials, to be quite honest.
So for those of us that are the hashtag we are not waiting community, the early adopters, the very impatient people, and I hear what you're saying, how do we get heard by the FDA? I've been trying to get to someone there, and I do understand the COVID global problem and emergency, but we've never had a whole lot of success in talking to them as patients. And even from the drug point of view, because we take insulin,
Right.
How do we do that? How do we be loud, be heard?
That is a great question. And I can tell you some of the things that we did when we were working with trying to get stem cells initially into patients. And again, I was on the periphery of this, but definitely was helping craft some of the strategy for it. And the approach was not so much to try to track down the FDA. That's kind of the top of the food chain. The best way that we kind of started with was partnering with companies, people at NIH, people who are going to be able to put this together. The FDA, being able to submit the paperwork for the FDA is a ridiculously formidable process. And having someone who has experience with it and a company that is willing to do it and has basically a baked-in set of advocates who are willing to enroll in this is something that I think is the best way. So what I would do is try to partner with companies who are doing the innovative research. And if it's you guys, try to figure out how to get a company on board with it, that they can, that everybody eats at the well. And I know that's not a very satisfying answer because that's going to take time. That takes taking into account things that are not really the pure help that is needed at this moment. And so it's really not very satisfying, but it tends to get FDA attention through companies. They listen to companies, not individuals generally.
So we have a question from the YouTube channel from Terry O'Rourke, and he's saying that we often see headlines based on preclinical trials, and there's a lot of hype that comes with some of these headlines about preclinical trial research. And I know even myself as a researcher, I have friends sending me articles like, have you seen this? And I'm like, yes, I've seen it. It's in mice. It's awesome. But this is not people. And we know that mice have been cured of diabetes over and over. So what's your opinion about this and trying to maintain hope, but also be realistic about these studies? Can anything be done to tone this down? What's your take?
I love this question for lots and lots of reasons. And Rachel, as you said, we've cured diabetes in mice countless times. And there's a lot that goes into this. And I think that this goes a little bit to what I'll talk about what we do at the DRC. And that is scientists are by nature not people who are as involved with the external side of science and promotion as universities. And I think what happens is that universities, in their attempt to put out splashy headlines that make them look good and are likely to bring in money to the institution itself, promote things in a way that oversimplifies. And I think that that's where the problem comes in, is oversimplification. And you'll see, as well as Rachel, as you know, science is really complicated. And if you try to take something and boil it down to its simplest element, you're going to wind up with something that's not just a platitude, but is also at its core incorrect. And this is exactly what happens with diabetes, with every other type of research. You see these exciting things that say, this can be the opening of the door towards, curing colon cancer. And yeah, it's a great first step. But nobody then thinks about the slides that we went through. That's at the very beginning of this process. I mean, we're talking here at this stage. We're not talking here. People don't understand that the basic research that looks promising is really going to take, in reality, another 15 years if it's incredibly lucky to do this. And the other side of this is that science largely takes place in cells outside of the body, human cells outside of the body, and mice. And neither one of those is really a very good model for what humans are. And so when somebody says something, what you need to be doing is thinking about this timeline here. And you need to say, this took somebody 2 to 4 years to generate. This is the beginning finding. What's going to happen after this publication is that if it's really worthwhile, drug companies are going to get on it. They're going to help license it from the academic labs. And then they're going to really start to look at how viable this option is. And so tempering what you believe based on a splashy headline is really kind of what I suggest that most people do. Do you have any suggestions? What do you tell your people and your friends?
Yes, I agree. When people send me things, I say, wow, this is really awesome. This is exciting. It took me 5 years to cure one mouse and it was a lot of work, but let's be cautious about timelines, and especially for the people with diabetes in their community. They've been told for a long time that a cure is around the corner, and I try to be very respectful of that history and say that we're working as hard as we can to get it as fast as possible, but I can't promise you timelines, and just be cautious about the things you read and look at it with a skeptical eye. But when things do end up working, we'll know about it, we'll hear about it, and we'll benefit from it.
Yeah, I get articles from people and they're very excited, and then I noticed the publication date was 2010.
Yeah.
Oh.
Well, that's another thing. So, and again, I'll get into this a little bit, because this was part of the rationale for helping establish Diabetes Research Connection, is finding credible sources of information and news about what's happening in the scientific community without this sort of hype and spin. Because basically I had a very small niche within the diabetes community. I mean, cell replacement using stem cells is far away from people looking at bionic insulin. And so I had to find where credible sources were. And so we try to keep up with that and try to help provide, real information that's up to date, that we think is interesting and has the potential to lead somewhere.
I think there are 2 more questions. Tina, you want to grab one on this whole process?
Yeah, so our friend Rebecca says that she frequently looks for trials that to participate in, and that very few are close enough for her to participate in. Why
Yeah.
are the studies locations so restricted, and why not open them up to more sites to get more participants?
That's a great question. And generally, it comes down to the problem of money. Now, let me also suggest that one of the places to look for trials, the best one within the type 1 community, is TrialNet. It's fantastic. And if people aren't using that, then they should definitely go there and look at that. That is an incredibly fantastic group of people who are very dedicated to the type 1 cause and being able to help people find clinical trials to participate in.
So I'm going to, 2 more questions. I said 2 before, but I lied.
That's okay.
Who volunteers? Do you have an idea of who volunteers for the first clinical trials? So I'm going to, 2 more questions. I said 2 before, but I lied.
That's okay.
Who volunteers? Do you have sorry, I had a mic on. Sorry.
Okay.
So
I don't know who volunteers.
What do they look like?
I have no earthly idea. I really don't. That's well beyond my expertise.
Okay. And I know this is going to lead into DRC because I've been reading a lot about DRC this week. Money. Let's talk money.
Yes.
So you come back to stage 1.
Mm-hmm.
How much money do you have to walk into with a drug, or is it dependent on how many people would be affected by the approval of a drug?
So is this going back to this idea of how much money it takes to build a drug? So there is there's been kind of at least in the drug world, there has been a push for curing orphan diseases, things that don't affect a whole lot of people, because they tend to be relatively straightforward diseases. You can think about a single mutation and how to repair that. So there has been a lot of money put into those efforts, and they've really started to pay off. However, when you get to something that is not a monogenic disease, a simple mutation of a DNA base, what you wind up with is something in diabetes that is really an entire organism, and trying to figure out the interaction between an islet within the pancreas as well as the immune system, it begins to get incredibly complicated because we don't have very good models for it. And so these tend to be much more expensive endeavors. Even though they have bigger payouts, they are much more complex and a lot harder to understand and deal with. Does that answer the question relatively?
Mm-hmm.
Okay.
Okay, so I said 1 more. Now it's a half question. So, and this is this very strange question, but what if we change the rules? What if we said, you don't need this depth of, so people die. But if you speed through the process, more people will live. Has the FDA built some amazing redundancy into the process that slows things down, and it doesn't have to be that way?
You know, this is more of a question for bioethicists, of which I'm not. It's a fascinating question of how to save the most and do the most good for people. And I don't have a very good answer for that. I would suggest this is a really good topic for a bioethicist. And, you know, for a long time, my friend Ted Friedman was involved with gene therapies on some of the FDA subpanels, as well as bioethics. And even though he's getting even though he's not as in science as he once was, I think he might be a good person for your group to talk to if you have questions about that.
Perfect. Okay, we'll let you go on now.
Yeah, thank you. All right, so I'm going to now switch gears as promised and move away from the FDA and a little bit towards science and what DRC does. And so I want to start by giving you what our vision is. And this is really what we have worked on and thought about for the past 10 years. And that is, we want to support innovative scientific inquiry until diabetes is eliminated. And really that fits well with what our mission is, which is, I think, very much in tune of how this group thinks. And that is, we want to connect donors, people who are, not, and by donors, I don't mean people who are just strictly money, people who have a vested interest in type 1, be it through family members themselves, whoever that may be, to connect these people with early career scientists that enables these scientists to perform peer-reviewed, novel research designed to prevent and cure type 1 and minimize its complications and improve the quality of life for people who live with this disease. So this was started by, and I'll get into all of this because we're now going back here just a little bit as to what it is that Diabetes Research Connection does, okay? And I want to go back to this FDA question. And as I was putting together these slides, that FDA question was just too wonderful not to use again. Because what you have here is this basic research, is preclinical. You have then the phases of the clinical trials and then the FDA approval. And what happens is, and I think most of you are aware of this, is that the basic research section here is this kind of phase where the scientists, both in academics as well as industry, are coming up with these ideas, actually starting to process them. And you think of these large funding agencies, the National Institutes of Health, JDRF, American Diabetes Association, as funding these. We fit in somewhere different. We provide the funds to test the new ideas, okay? So this is an important part of exactly how I think science should work. And if you disagree, I would love to be able to have this discussion, which is people don't need a lot, people who are postdocs in well-established labs don't need an awful lot of money to be able to test their ideas. Generally, they are just starting out. They've got things they want to test. They have plenty of the resources. And so what they need, though, is a little bit of time and some money to be able to say, I love what's been happening, with X, Y, and Z in the field. Let me go off and try to see if I can do something much more dramatic to help accelerate this. So what DRC does is we are the absolute beginning of this FDA pipeline. We aren't trying to fund clinical trials necessarily. We aren't trying to do anything through all of this. What we want to do is give people the time and money to be able to test what their idea is and whether it has legs. And quite frankly, all we're interested in is the data. Does it work or does it not? And if it works, man, that's great. Then go to the NIH, the JDRF, ADA, begin to get these larger funds, begin to test it, get it into this pipeline. But if it doesn't work, we're also interested in that. We want to be able to say, hey, this idea or this technology or whatever it is doesn't work. We as a field need to move beyond this and start thinking about something else. So the failures are equally important as the successes in this. So this is where we fit in. So I'll take a break and see if anybody has anything to say at this point.
So we are wondering if you could give a background of the DRC.
Oh, that's
how did it start?
I will give all of that on the next slide. All right.
Yeah, a question, what else, who else, is this what happens at university levels? Is this?
So this is normally happening at the university level. We have had some people from kind of hybrid, sort of affiliated with universities, but kind of affiliated with private companies also do things. But we tend to focus on the academics because the academic tends to be much more nimble than the company. The company seems, has lots of things going for it, including shareholders, bottom line, stock prices, all of this sort of stuff. Academics sometimes just have the idea. And so that's where we've tended to focus. Okay. So
absolutely. Are you working at all with the sugar science?
We have helped them get started. They kind of came to us for some advice. They have an interesting model, and we are definitely in contact with them.
Got it. Thank you for doing what you do. Any other? Go ahead.
All right. So this is the DRC, what we do, how we got started slide. Okay. So this started about 10 years, almost 10 years ago. And I'll point you across the row to the people who were in the room as we got established. Unfortunately, you've had to look at me and listen to me. Let me introduce Nigel Calcutt, Amy Adams, Alberto Hayek, and David Winkler. These are the people who helped found this organization. And this really came from David and Alberto having a discussion about what's going on. How can we help accelerate research? How can we get funding into the hands of the people who are really testing these new and fascinating and amazing ideas? How can we get them that money, that seed money, to be able to begin to do the research that we think will lead somewhere? So over the years, we have been fortunate enough to pick up many board members along the way. Steve and Bobby and Paige, our new scientific director, Vincenzo, and Felice. And down at the bottom is our very amazing and able staff, led by Karen. And then there's Casey and Hannah, who many people on this have dealt with. And so we really wanted to do something different in this, okay? And as we discussed this, we realized that exactly these problems with these long timelines were problematic. And so we said, why don't we, if you sat down 50 scientists in a room and you gave them a single piece of paper with an idea, it didn't have to be their, it didn't have to be in their field, as long as it was well written and understood. Those 50 people, you could get a majority to say, let's give it a shot, or maybe not. And these are people who are at, the highest ranks of academics. Alberto and Nigel and I are lucky, mainly Alberto, to know people all across the country who have agreed to donate their time to come in and read these one-page applications from these young scientists. And by young, I mean just starting out. And what they do is they read it and they say yes or no. And it's 50% plus one. And if they say yes, then what happens is we then pick 3 people out of that board of it's now about 80 scientific reviewers. And we pick 3 people who are experts in the field. So if it's an immunology one, we pick immunologist. And we tell the person, we say, hey, this is a project on this. They're going to write 3 pages, only 3 pages. For those of you who know, NIH grants can be incredibly long, 20 to 30 pages. And for certain program project grants I've had with supplemental figures and publications added, I've had them go over 1,000 pages. And so this is 3 pages. And it says, here's what I want to do for a year. And $50,000 to get us to the point where we can say go, no go on this. And those 3 reviewers basically say yes or no. And if they agree, then it goes up on the website. And this is really one of the things that I am most excited about. I've talked about the rapid, and I've talked a little bit about the peer review, but I like transparency. And this is really fascinating to me. And this is really something that I am very proud of and I love very much. And that I think this is something that separates us from everybody else. And that is, if you give money to basically any charitable organization, you have no idea or very little idea of where your money actually goes. With DRC, if you give money to a project, not only does 100% of your money go there, but more importantly, you find out what happened to it. You get a 6-month update that says, here's where I am in my progress. And then at the end of it, you get a final report. It's published on the website. It says, here's what we found. You actually get to see what it is that your money does. So we fund people from all across the country at, academic institutions from UCSD to up in Harvard, from Florida to Seattle, all across this country. If there is a diabetes grant that has merit and has, strong support, it will go up on the website and hopefully be funded. Now, we've traditionally used a crowdsource model to fund this. We've recently switched somewhat, and that any grant that goes up on our website will be funded. And we work hard on the back end to raise that money, not just from crowdsource, from anybody who donates, but also through other donors who have the same vision as we do. So I'm very, very proud to say that with this last round, it looks like we will be able to fund our 50th grant this year in our 10 years of existence. And we will have donated, this is a little bit outdated, but well over $1 million to research. And then really what is also critical in all of this is what's the impact? What's the return on investment for this? And so what is the impact of a $50,000 investment? And so here are a few examples of people who have taken this money and gone and entered into this pipeline. All right. And they've gotten the NIH, the JDRF, the ADA grants. And you can see here that Todd Brusco got $500,000. Wendy was able to get $4.5 million. Joseph Lanthman got $1 million. Peter got $500,000, and Yo got $1.2 million. The average return for every dollar that's been donated to DRC projects is a return of 30. And I am incredibly proud of that. So at this point, this is a nice point to stop to answer questions. I know Paige, I can see, is on the call. That there's people who are on the board of DRC who want to say something that I missed or want to emphasize something that I may not have emphasized enough. I certainly welcome their opinions, but I also welcome your questions because we're going to get into some science here soon.
Yeah, I was going to kind of shift you towards science because we want to kind of focus on that. And we know you raise funds, duly noted. So, but we would love to know more about the projects that have been successful. Do you have a favorite? Has there one that has just gotten legs and gone gangbusters?
So there are a number of them that have done well. I think one for the patient side that was a very early funding of ours that I won't talk about is CGMs, continuous glucose monitors, as in hospitals as basically a standard of care, and have people have their glucose monitored just as part of the regular sort of check-in within the hospitals and how that worked. And that seems to have worked incredibly well. I think Joseph Lantzman's work has been rather fantastic, and he uses a non-human model of to look at how stem cells can transdifferentiate directly into insulin producing cells. So in other words, have muscle cells be able to make insulin. So these are a few of them. There have been a lot of people who have been trying to generate different types of insulin, looking at beta cell senescence. So we're going to get into a few of these that I like. And I'm going to talk about some ones that are projects that are currently being offered that I think are quite interesting. So if we're ready to move into that, let's go. All right. So this is one that I've really enjoyed. And I'm going to basically run through this, and this is going to get very scientific very quickly. I'm going to try to save a lot of high-level stuff. I'm going to try to keep this at a high level, but if we need to get into the weeds on this, or conversely, if you don't understand and have additional questions, I'll try my best to answer, okay? So one of the ones that's currently moving now is, will understanding the function of a specific gene in a protein reveal the cause of type 1? And this is again by Yi Wang at UCSF. And so the idea behind this is that you have something called thymic medullary epithelial cells, MTECs, that are normally, these cells train T cells to recognize the body's own protein, okay? Now, insulin is one of the several tissue-specific antigens that's expressed by MTECs as part of this training, okay? So again, you have this MTEC, self-reactive T cells, expansion, autoimmune disease, okay? That's how the idea works. So insulin expression depends on this protein autoimmune regulator called ERI. And ERI prevents autoimmunity by exposing T cells to TSAs, these tissue-specific antigens, and destroying those that react. The elimination is negative selection, okay? So you're getting rid of cells that are to the self. Now, ERI expression in MTECs represents a clinical barrier and a breakdown of this T cell education. In both humans and mice, ERI deficiency results in the failure of negative T cell selection and multi-organ autoimmune disease, including T1D. So in other words, under the normal thing is you get the ERI expressed, and the regulatory T cells are able to respond correctly. So what is Yi thinking about? So here's a picture of the ERI protein, and you can see that it has all of these various domains within it, PhDs, a SAN domain, a spectrum domain, and I've colored the amino acid sequence according to these domains, okay? And ERI falls into a class of proteins that are called transcription factors, okay? And transcription factors are proteins that mainly live in the nucleus, but travel around between the cytosol and the nucleus. And what their job is they sit down on specific sequences of DNA and they turn the gene on, okay? So gene turns on. So that's what ERI does. And I don't want you to walk away thinking that transcription factors are simple DNA binding. It's always a lot more complicated. There's distal elements, there's activators, there's mediators, there's polymerases, there's promoters, proximal controls. This is really, really complicated stuff. So the transcription factor is really just one protein in probably 10 to 50 proteins that actually regulates whether a gene gets turned on. So in this, Yi said that their preliminary data provides strong evidence that this PhD1 domain plays a critical role in regulating islet-specific, tissue-specific antigens, okay? Therefore, they hypothesized that this domain of ERI regulates a unique set of genes. In other words, it binds to and activates and turns on a number of genes which are critical for negative selection in islets. So what they're looking to do is figure out exactly what these genes are, then figure out how they can take them a step further and determine whether ERI can be regulating those either in an on or an off way. So that's one of the projects. Feel free to ask questions about it, say you didn't understand it, say that I was too simple, however you want to go in.
Probably a bit complicated.
Well, you wanted science. So what can I do to help try to make this a little more understandable?
I was wondering if you could mention what kind of processes they're using to investigate this. Are they working with animal models? Are they working
So this
with...
is an animal model study. This is
okay.
a great question, Rachel. So this is an animal study. And what they have done is they have made a mouse with a PhD knockout, and they've made another mouse with this sand knockout. And the sand knockout mouse does not have the same phenotype, which is appearance or outward sort of characterizable sort of, how would you call it, phenotype in simple terms? Basically what the thing looks like and does, whether it's sick, whether it's healthy. The knockout of this is not the same as the knockout of this. Only this is the one that plays a critical role in regulating the islet-specific TSAs. So what they think is that this protein here, it has multiple interaction partners, and that if you disrupt one of these interaction partners, be it an activator, a mediator, some distal control element, that you are not getting this negative selection that you would normally get. So they think that this is the key. Now, what are different ways one could possibly approach thinking about this? And I'm going to say I'm completely off script on this. I don't know what Yi is planning to do beyond all of this, but one of the things one could do is take this little piece of protein and express it itself and just see how the landscape of gene expression changes. So in other words, you're able to go in and manipulate this one protein, this one domain, probably one or two amino acid changes within this, to be able to select whether you can revert this negative selection loss, right? Because really what you want to do is make sure that the T cells don't recognize insulin as something foreign, that they recognize it as self. So in other words, you can think of this as retraining these cells to do what they should have been doing at the beginning. Now, is this a much larger piece of a reprogramming strategy? Of course it is. But this is probably a pretty important one to understand. If this is important in training, finding out what it's binding to, who it plays with, and what time it plays with it is probably going to be essential for understanding what's going on.
So if their hypotheses ended up being correct and what they think is going on here, what would the therapy look like? Would it be some kind of injectable? Would it be a candidate for CRISPR? Do we have an idea of that yet, or still early days?
I would say that this is still so early in the pipeline. So
okay.
Next one that I'm going to talk about is something that we can maybe together think about what a pipeline looks like. But I specifically chose this one at the beginning because I wanted everybody to get a picture of how this is a complex, not just beta cell specific disease. This is something that involves lots of different types of cells interacting throughout the body and everybody thinks, oh, it's the beta cell. Oh, there's this idea of how the T cell comes in and destroys it. There's a lot more to this. The T cells under normal circumstances are trained not to recognize. And this is an avenue that has not been well explored in this. How is the T cell trained to not recognize the beta cell as foreign? And so it's not an idea that a lot of people have been thinking about. And that's why it's something that we were excited about. Because if you can retrain a T cell, then that starts to get at the root of the problem. All right, shall we move?
We should move.
All right, the next one is talking about old cells. And this is pretty interesting to me because this has really become an emerging field within not just beta cell biology, but in a lot of biology. And this is will understanding why normally good but aging beta cells accumulate with the progression of T1D, and will it unlock the mystery of the disease? And this is Yao Wang again from UCSF. So one of the things that happens under normal circumstances is that you have your normal setup of pancreatic islets here, and it's made up of these beta cells. And then with time, insulin resistance, all sorts of other things, you wind up getting something called a senescent beta cell. And so this is a cell that basically is old, might not function like the young ones, and is really characteristically and fundamentally different from a properly functioning beta cell. And so what happens in these senescent cells is that the identity of the beta cell genes goes down. You lose things like MAF-A and other things that make these beta cells beta cell-like. And you then upregulate a couple of different things, something called SASPs, which we're going to talk about. And what that tends to relate to is beta cell function and proliferation. Both of those decrease. Now, SASPs are senescence-associated secretory phenotype. In other words, what these senescent beta cells do is they spit out little vesicles with proteins, RNAs, microRNAs, all sorts of little packets of information, if you want to think about it, that tell the cells around it what this cell is doing. And these cells that are healthy and normal around it take it up and incorporate this information. They're learning. And this information isn't always the best information for them. If this cell says, oh, I'm really old, I'm getting ready to die, this other cell thinks maybe the whole organism's going to die. So understanding what happens in this. And so there's this idea called senolysis, which is pretty cool, which is getting these old cells to just kill themselves. And when you do this, you're able to induce beta cell identity genes. You lose these senescence-associated secretory phenotypes. And the question is, do you see more beta cell proliferation? And do you see more whole body insulin sensitivity? So again, here's the idea. You have this senescent cell that is releasing these little small SASPs, and these healthy cells are looking at it. And what's happening is you start to get, within these things, you start to get these stressors of cells that are built up from DNA damage, these cell cycle regulators, oncogenic activation, telomere dysfunction, reactive oxygen species, insulin resistance. All of these things are encapsulated in these little vesicles. So what is it that Yao is looking to do? So here we have extracellular vesicles which carry and transfer various proteins, RNA, and lipids that are derived from the parental cells. These extracellular vesicles function as carriers of signals that regulate immune responses. So in other words, beta cell, you have impaired function, you then get improved function if you can lyse these beta cells. These contain things that are associated with upregulation and recruitment of immune cells, T cells, macrophages, monocytes. So they might regulate, so these senescent beta cells might regulate the immune response via the release of these extracellular vesicles. So what they want to do is to determine the role of these that are secreted from senescent cells and develop and see how they relate to type 1 diabetes. In a previous project that we funded, what we found is that Peter Thompson was able to look at and develop chemicals that are senolytic compounds. And so this is kind of an idea of how beta cells themselves might play a role in furthering their own death by bringing on and recruiting immune cells to the pancreas and to islets. Does that make sense?
Yeah, yes. It sounds to me, I'm not a scientist, but it sounds to me like beta cells are kind of smart little fellows.
All cells actually are pretty smart. And, you know, I like to think, so if you think about the cells themselves, they don't really have a lot of information if they're sitting in a dish. But when they're in an organ and in a functioning animal, they are able to, or plant, they are able to contextualize what is happening in the system. So it's the idea of saying, I've got a beautiful car, and not taking into account how the road plays a role in how that car runs. So think about everything outside of the car as the road. And the road is an equally important contributor in all of this. And so what happens is these beta cells, as well as everything else, are continually spitting out information and proteins into the extracellular media, into this sort of road work, to be able to give cells information that they can use as to what's happening around them. So they aren't necessarily smart as in, we know we can do things. What they're doing is they're taking the information around them and using that in a really smart way, and understanding how that plays a role in the development of type 1 is absolutely critical.
Interesting. There was a question, Brad, always interesting questions. What about thymic education of new immune cells? Therapy would need a TP, I don't know. What about thymic education? Need to be, I see it, need to be ongoing, or is there thymic education that's altered permanently?
So that's a great question. And that delves pretty far into immunology. And I'm not an immunologist, but I can tell you that from discussions I've had with people, there is a lot of emphasis that is starting. Really, it's been over the past couple of years in re-educating cells to be able to do very specific tasks. So, you know, this all started with the immuno-oncology, which is trying to get the immune cells to go in and recognize tumors as foreign and be able to take that and use the human's immune system to actually destroy tumors. And so people are going about this now and thinking about this in the context of autoimmune diseases. So yes, this is going on. I cannot tell you the larger implications or ramifications of it because it's not my field, but I'm certain that there's somebody out there who could. And if you're interested in me finding out, I'd be happy to.
Thank you. So moving on, there was one project that we had talked about having to do with the different types of diabetes. And I think a lot of us always wonder, what kind do I actually really have, and how many kinds are there? Are you prepared to talk about that one?
I'm not, but I can tell you that I'm pretty certain that everybody's type 1 is different. I firmly believe that no one has the same type 1. I have seen people with, I got a call a number of years ago from a woman whose son had tragically died in a motorcycle accident. And she said to me, I think that some institution needs to look at his pancreas. He had type 1 for, 30 years. And I was like, yes, without a doubt, it needs to get to the right people. She said, but there was something strange about it. She said, every year for 2 weeks, he would take a vacation from type 1. And he just didn't take insulin, knew the right diet to have, and lived just normally. And I said, for 2 weeks. And she said, yep, 2 weeks. And I found that to be absolutely fascinating. And, against what I had thought everything that type 1 was. When I got involved in type 1 because of Alberto. And then subsequently, after about 5 or 6 years into my work on type 1, my close cousin was diagnosed, and she was 32 at the time. And her type 1 is, a very strange type of type 1 as well. And she's able to do a fantastic job of controlling it, but it's hers. And when people discuss diets and what she does and how she lives, people are like, but I can't do that. And I'm like, no, most people can't. And so I would say that based on the people I've talked with throughout the country for all these years, everybody's type 1 is different. And everybody knows and understands their body. And those who take the time and effort to be able to really understand what it is about them that makes it different. Hold on one second. To understand what it is that makes their situation unique, really does well. I'm sure you guys could tell me stories that would absolutely fascinate me all night long about what is unique about you. But I would say that the different types of diabetes are that you're going to continue to subdivide these categories. And to really get at your question, I think that when the day is done and there are treatments available, that there's going to probably be somewhere between 3 to 10 large buckets of what most people fall into as far as treatments go.
So when you say everybody's is different, are you meaning that the biological level, like what's actually happening in their
I do.
body?
I mean, at the biological level. I mean, so people who have type 1 still make beta cells. You are still making beta cells at a, regular level. And there's a number of functioning beta cells, but it's a cliff type thing. But the average person has 100%, but you can actually live with anywhere from 70 to about 30% of your beta cell population and still be asymptomatic for type 1. It's once you cross a certain threshold and you can't get back over it, that is where the problems begin and where really the effects of type 1 kick in.
So I know you guys have a tremendous wealth of your projects and historical wealth, and I'm watching the clock because we got people all over the country watching. Is there a way that people can sign up for an updated newsletter to know what's going on there? Because your projects are interesting and different, and if anyone's interested in specifically delving into one.
Well, let me finish with a couple more quick ones, and then I'll show you how to contact us, and we'll go through these pretty quickly, and I'll keep it incredibly high level. Okay. So we have one on diabetic retinopathies. And Joanne, I will also send you these slides if you want to distribute these. I know most people have them and will have this. So we have, this is actually a pragmatic approach within Johns Hopkins to use AI to look at diabetic retinopathy in children with T1D. It's a fascinating project, and one that I'm very, very proud that we are supporting. I will go through this next one very quickly because I have a different reason. And this is looking at hypoxia, which is lack of oxygen. And so this goes, the reason that I included this, and again, I will show more than this, I'll move on to everything else, is that there was a large hope for many years that transplantation, both of islets as well as stem cells, were going to be tremendously useful therapeutically. And I still believe that they are. But one of the main problems with it is hypoxia, which is the lack of oxygen. And beta cells are incredibly sensitive to this. And figuring out how to get cells that are both able to have access to nutrients from innervation and growth of blood vessels into them, as well as oxygen, is very key for keeping these cells alive. And Dr. Ash at UCSF is looking exactly at these things, how to take these populations of cells and make them stay alive better and longer so that engraftment and encapsulation will provide people with a better opportunity to keep more cells alive. So, one other one, which is the bionic insulin. I figured you guys would be interested in this one. I've included here a link, and this project has been finalized. And so I encourage everybody to go to the website to read the results. Again, this goes back to one of the unique things that DRC does, which is it provides you with information as to what the results of your money have done. So, and let's see here. This is hypoxia. Okay, so this is how to connect with us. Excuse me. And so we are on the following platforms, but it's really easiest to just go to diabetesresearchconnection.org. And I'm happy to take any other questions and happy to talk with people. Info at diabetesresearchconnection.org will get you to somebody who will find you, who will find their way to me.
Well, yes, thank you. David gave us the link and Rachel as well. I think there's a level of excitement thing. There's stuff going on. And for those of us in Southern California, there's stuff going on locally. So thank you. I will make sure the links are included, and we would love the slides because I will share that with the community. Is this something that those of us in the looping community could share with our medical caregivers that might spur some interest for clinical trials?
I'm all for anybody doing anything they can to bring more clinicians into this, to try to connect the people who see patients with the people who are doing the research. I think getting these groups talking to each other does nothing but benefit everybody involved. So I have zero problems with it. And if they want to contact us and become involved, become a scientific reviewer, do anything, we welcome any sort of help we can get.
That leads me to a question I had, and I may have just missed it when you said it. I remember you were talking about people volunteering to review proposals, and what are the criteria for that?
What we do is we've got a great lay review committee, and what they do is they work with the scientists and us to try to take this and translate it into something that people will understand, because part of this connection is taking and deconvoluting some of the complicated science into bite-sized pieces that people understand and can get excited about and can get behind. So if you want to, we'd love to have you guys.
What do you do to get involved with that?
Just contact DRC.
Okay.
That would be great.
You know, I'll share all of that and give you I'll give you some emails. Thank you. I know we've been trying to do this for a while. It's exciting, the work that's going on, because it's not things we talk about on a regular basis. So thank you so much. This will be available, and we're going to spread out the recording so others can see this later on. Thank you, co-moderators. Thank you for DRC, who's on the call, for joining us. Thank you for doing what you do to make our lives better. We so very much appreciate that. Thank you.
We are all in this together, and this is all of our love and passion. And so everybody on our board is in some way related to type 1, whether they haven't or family members or have been involved with people. So this is done for the love of it and the desire to see everybody get better.
Thank you. Thank you so very much.
Thank you.
Yeah. Good luck with your painting. Your dogs are adorable.
Yes. Thank you. Glad the dogs weren't too bad today.
All right.